So, welcome everyone. My name is Sara Evans. For those who may be new here, I'm the Senior Manager and Curator of the Living Collection here at Greenwood. And I do, with my job, get the honor of assisting and supervising research here related to the natural environment. So last year, Greenwood launched a small grant program for emerging investigators. Sorry, I'm just going to admit a couple more people. In the field of urban ecology. And the two awardees are here tonight to briefly showcase their projects and discuss the driving scientific questions behind their research, the data that has been collected and analyzed so far, and of course, they can help answer some of the big questions of why does this matter? Why do we care? So first to present is going to be Myles Davis. He is a master's student of ecology at Columbia University, and he mapped and assessed raccoon distribution across Greenwood. And then following Myles is Dr. Theodore Muth. He is a biology professor at Brooklyn College, and he will be discussing his survey of soil microorganisms across Greenwood. But before I hand the screen over to Myles, I'd first like to lay down a couple room rules. Number one, please keep your microphones muted during the entire program. There are so many of us in here. And number two, do not hesitate to ask questions and share comments. Just drop them in the chat box. I will be keenly monitoring them and meeting them to Myles and Theodore. So with that, Myles, take it away.
Thanks, Sara. Okay, I'm just going to share. Okay, how does that look, Sara? Okay, perfect. So, hi everyone, my name is Myles. I'm a Brooklyn native, wildlife biologist, and a master's student at Columbia University. Today, I'm going to talk to you about my transect that my collaborators and I have created to examine mesocarnivore distributions across New York, and my work examining raccoon distributions within Greenwood Cemetery. So the work I do falls into the field of urban ecology, the study of urban ecosystems and wildlife in them. An emerging concept within this is reconciliation ecology, which is how we can preserve and increase biodiversity within urban landscapes. Reconciliation ecology has started to gain traction as we realize more and more that cities can accommodate various wildlife species. And this concept is supported by phenomena like wildlife climate refugees and the recent appearance of wildlife in cities following COVID lockdowns. So these cases have drawn our attention to the potential benefits like pollination and conservation, as well as the risk like disease spillover and property damage of increasing wildlife presence in cities. So in order to better understand the potential for these benefits and risk, we need to study what factors influence wildlife distributions around cities. And this leads into my broad citywide question, which is how does urbanization in New York City relate to mesocarnivore distributions across its green spaces? So I'm going to break down some of these terms. Urbanization is the process of making an area more urban. It's associated with changes in infrastructure, land use, and human population. And it's generally thought as we go from more rural to urban landscape, we find less species. And since these urban areas have higher density of people, this can lead to more human interaction with the wildlife that do remain in cities like mesocarnivores. So mesocarnivores are mid-sized mammals that have a more generalist diet, and they're able to take advantage of human-associated resources like leftovers and trash, and they tend to reach higher densities in urban environments. There's conflict that can arise from this, like conflict with pets, property damage, disease transmission. But that's not all to say that mesocarnivores are bad for cities. There's a lot of benefits as well. They're scavengers, so they remove decomposing animal matter, and they're predators, so they can control pest populations. And below, I just have some common mesocarnivores that you might see in New York City. But within urban landscapes, green spaces are places of interest for researching these taxa. Green spaces like Greenwood Cemetery are natural to semi-natural ecosystems around a city or just anywhere with grass in the city. And because they're often used by mesocarnivores for foraging or nesting, we can use them to get an idea of which species are present in the area. So now that we've gone through the terms, we can revisit the question. And along an urban gradient, we can sample different green spaces to see what's present in the area and what level of urbanization do certain species tolerate. And this allows us to better understand the urban wildlife conflict and conservation landscape. Specifically, I'll be looking at how species occurrence, which is whether they're there or not, and occupancy, which is the probability that they're there or not, how that changes from Brooklyn to Long Island. And I'll show you an image of this. There's a lot of different ways to measure urbanization. You can look at it like housing density or percentage of concrete or tree cover. And again, I'll show you what some of these variables look like. But part of answering my question is understanding which aspects of urbanization best explain the distribution of these wildlife. We're also incorporating phone contact tracing data that we can use to get accurate measures of actual human visitation in the green space. So this data set was developed to address COVID safety, and it hasn't really been used for wildlife studies yet. So it's an interesting opportunity to see how actual human foot traffic affects these distributions as well. An analysis across 10 North American cities found that several mesocarnivore species, they show a negative response to urbanization in cities with high housing densities like Denver and Chicago. So as you get closer to the city, as you can imagine, you find less of these species. And as a high density city like New York, we'd expect to see the same thing. They couldn't really draw any conclusions for skunks. And interestingly, while coyotes also do show negative responses to urbanization in this 10-city study as well, the response did get weaker as a city's housing density increased. So it'd be interesting to see in New York, especially what this relationship would look like, since New York City has some of the highest housing densities within the country. So we're using camera traps for our sampling. I actually have one right next to me here too. And they're ideal for this kind of study because they're cheap and they offer 24-hour passive monitoring. And how it works is whenever something walks by, boom, it just takes a picture of whatever is walking by. So I'll show everyone an image of the transect that we have. And so this is our study area. Our transect runs through Brooklyn, Queens, and Nassau County. And it's about 50K. And I'm going to show you, so this is an example of what an urbanization gradient looks like. And I'm representing urbanization with percent imperviousness. So that's basically the percentage of concrete. And you can see we go through urban, suburban, and exurban land. And these black dots are each a camera site. And you can see the beginning of our transect is actually Greenwood. So here are some other maps just showing what variables we can use to represent urbanization. This is, again, the concrete. We can use tree cover, housing density as well, but we're always following that urbanization gradient. And just some images. So the actual setup of the transect involves me and others physically going out to all of these green spaces, talking with landowners and setting up the cameras. Greenwood Cemetery. We have a combination of cemeteries, golf courses, parks, gardens, and some smaller city parks. It's not just big, lush green spaces that we're using. Some of the images that we've already gotten so far. Coyotes. Yes, there are coyotes in New York City. People might have seen like New York Times articles, but yeah, there's coyotes in New York City. This was on the edge of Nassau and Queens. Possums are everywhere. Most people think they're big rats. Striped skunk. This was in Greenwood. There's a lot of skunks in Greenwood. Fox. This was on the edge of Nassau. Some cool deer photo. So this one has a nice white color morph as well, and raccoons everywhere. So this is the start of New York City's first long-term wildlife monitoring. Transect is a huge collaborative effort. Some details about the data collection: we collect January, April, July, October, and we started April of last year. And while we're only in Brooklyn, Queens, Long Island, there's plans to expand to Staten Island and the Bronx. And once we're only in our first year, but once we have collected more data, we can do predictive modeling to look at how species distributions might change with future urban expansion and with more regreening as well, which is important for city planning. The cameras also detect more than mesocarnivores. They can detect any kind of mammal, really. And data like this has been used to kind of see how deer presence correlates with tick abundance and Lyme disease prevalence in the area. And there's plans to use this transect data for that question as well to gain further insight. One of the more obvious benefits is that looking at how population distributions change over time. I'm sure a lot of people who have been in New York a while will say that there was never really like raccoons and other wildlife around when they were younger. And that's changed now, but we don't really have any data to prove that. 311 calls are the most organized database for the raccoon population. So this is the start of keeping track of that. And this is all nested within a much larger organization, the Urban Wildlife Information Network, which we joined last summer. It's a camera chat network in over 40 cities doing studies similar to this. And by joining this network, we can compare our study to cities across the world. So on a smaller scale, I'm also interested in raccoon distribution and space use specifically within Greenwood Cemetery. So my second question is, how does raccoon space use vary within Greenwood Cemetery? And why am I focusing on raccoons? They are the most abundant mesocarnivore in cities across the US. They're also the most reliant on human food sources. They pose the largest risk for rabies infection, but they're also important for predation. They eat wasps, and they're also important for seed dispersal as well. But they basically represent kind of the largest source of mesocarnivore conflict and benefit within cities. And there's active rabies management going all around New York City currently, and it's always a management concern in areas like Prospect Park and around Brooklyn. To the right, it's a picture of raccoon feces. Raccoons like to poop in the same spot, and they're called latrines. Like 10 raccoons will all poop in the same spot for weeks. And some of them carry a raccoon roundworm that can be potentially fatal if left untreated. There's only been 30 human cases since the 70s, so there's not a lot of info known, and the risk to humans isn't that great, but there's a huge risk to wildlife. It's anything from a larval migraine to something like paralysis or blindness or even death. And it's present in New York City raccoon populations, but that's kind of one of the reasons why it's important to understand where raccoons are congregating, because that's where you might find the most poop. Okay, so detection of raccoons and their space use varies even within a 1 hectare plot. And Greenwood is almost 2 hectares. So chances are raccoons are not using this space equally. It's been shown that raccoons preferentially select habitat that's forested, as well as habitat that's associated with human food sources like trash cans. Their presence has also been shown to increase near water sources, as well as edge-like habitat. So where green habitat meets residential areas, because these human-dominated areas actually have more food sources. So my question is, how does raccoon detection vary within Greenwood Cemetery, and what factors may be influencing this detection? So there's distance to edge, distance to trash, distance to water source, and tree cover density. To answer this question, I've created a 40-cell grid within Greenwood Cemetery, and these cell sizes are based on the home ranges of a Greenwood raccoon, or a few Greenwood raccoons that were radio collared by a previous PhD student. And you can think of each of these squares as the size of a raccoon's home range. And the cameras are 100 meters apart. They're all facing north and at knee height, so everything's standardized. And each camera, I calculate those variables I told you about: distance to edge, distance to a trash can, tree density around the area, distance to water source. So the cameras were put out in the summer, 2 weeks. I set 20 of the cameras out for 2 weeks in June, and then another 20 in August. I did this because I didn't have 40 cameras, so 20 at a time. And the intended outcomes are a wildlife photo database for Greenwood. So, there's a project website for this where locals or interested parties can request permission, gain access to some of the photos. Also, a raccoon detection heat map highlighting potential hotspots for raccoon activity within and around the cemetery, as well as identifying significant sources of attraction within and around the cemetery. So the grid data is still being analyzed, but here's some data from 2 of the transit cameras that I've had out in Greenwood from April. And as you can see, raccoon detections are way higher than any other species. And to the right is just their daily activity trends. As you can see, their activities focused around night and early morning, and so little overlap with humans. So the outreach part of this is very near and dear to me as someone that grew up in Brooklyn and very curious about wildlife. All the photos from our cameras, like I said, are put on a citizen science website where people can request access. And what's really nice about this website is it filters out all human photos, so it's only the wildlife you see on there. And it's been rewarding to promote the transect to public forums. People are really excited about the chance to see what's in their local green space. There's also been a lot of student involvement. And last summer, we actually had the New York City Girl Scouts come out to Greenwood, and we had some news coverage as well. And they were helping set camera traps. I'm also currently working with a middle school in Brooklyn, and they're going to use some of the data for their own research project and go on a Greenwood field trip in April, hopefully. Oh, next page. It's a little slow. Oh, there we go. So overall, both of these projects have management benefits. So firstly is improving wildlife awareness around New York. All the photos I showed you of mesocarnivores were taken at night. They're elusive and nocturnal, so it's really difficult to know where they are without 24-hour monitoring. And in New York especially, there's a huge knowledge gap here. We don't know what's around, and we're really surprised when something turns up and we get these kind of headlines. The distribution data can also aid with disease prevention, like knowing where to place oral rabies vaccination baits for raccoons for citywide vaccination efforts, or within larger green spaces like Greenwood. And this can help identify key conservation areas within the city. Since 2007, we've seen the return of beavers, otters, and coyotes. So this information is becoming even more relevant now. And yeah, this is all part of a huge collaborative effort. So I want to give thanks to Columbia, the Duke Wasser Lab, and Gotham Coyote Project for donating cameras, Greenwood Cemetery for granting us funds and connecting me with journalists, and all partner organizations for keeping this going and making it all possible. Okay, thank you. And I think we'll take some questions now.
Yeah, that's right. So a little bit. There was a question if each image that was captured, each animal, if that is a, is there redundancy in there? Are you potentially capturing the same animal twice, three
Yeah,
times, four
that's a
times?
great question. That's a great question. So sorry, my computer's a little slow moving back. So with the Greenwood grid and the transect, we have a certain distance that we maintain between cameras. So for the transect, because the species we're looking at is anything from a deer to a raccoon, we have 1,000 meters between each camera. And that is so we can assume each camera is independent. So a camera, let's say a raccoon comes up on a camera in Prospect Park. That is not the same raccoon on a different camera, and that's because they're that far apart. The same for the Greenwood grid, because it's only raccoons we're focusing on. They are 100 meters apart, and that is so that we can assume independence. And you can also see from the grid I created, each of those is go back to that. Each of those grid cells is based on the home range size of a raccoon, and a Greenwood or New York City raccoon. And so that cell is kind of sorry, it's all wacky. The size of that cell is the area that a raccoon might actually just stay in and not move out of that. Yeah, obviously, there'll be some differences, but this is based off of actual GPS tracking we've done on a few raccoons.
Joe says, bravo, Myles, like 8 times. Um, so, James, we have not found any foxes in GW yet. Nor have we found any coyotes or evidence of coyotes. So a couple other questions that Myles, you and I may be able to answer. The rabies mitigation efforts, what does that look like in the city? I know that at Greenwood, we connect with the Department of Health and Mental Hygiene, and they put out, they're like these PVC pipes that are just filled with these little packets that look like ketchup packets, but they're actually an oral rabies vaccine. They
Yeah.
supposedly taste very delicious, and so they attract um, skunks and raccoons to eat it and become vaccinated against rabies. And
Yeah,
then,
that's right.
yeah, um, that is definitely something that we do at Greenwood as well. Do you know of any other sort of mitigation efforts?
No, I would say that's the main thing, the rabies vaccination, the oral ones. See if I have a picture here. I
You have
believe like
the packets. And then
they look different. Yeah.
A. Logan wants to know if you have any thoughts on whether skunks will eventually appear in Prospect Park or if you have captured skunks on your cameras in Prospect Park.
Wow, that's exciting. I hope to. It's possible. Skunks are very finicky. And based on the reading I've done in other cities, they love cemeteries, and I guess maybe they like to burrow, and there's already like underground systems within cemeteries, but um, it's very possible for them to come across to Prospect Park. I haven't gotten in on any camera traps yet. Prospect Park staff have seen a skunk before. So if you look on Prospect Park's website, mammals that have been spotted there, you'll see skunks there. So yeah, they're right next to each other. So it's very possible.
That's awesome. And then do you know how has urban living influenced raccoons' lifespan, their nutrition? Jada wants to know if they live longer or have more offspring in urban systems.
Yeah, that's a great question. I don't actually know about lifespan is I can tell you they definitely get they get bigger here. They have a lot more food sources here, and you know, they are so well supplied from our food sources that they will ignore their natural food sources for ours. Like they'll stop eating like eggs or other insects because our trash is so delicious. There's a lot of different causes for raccoon death within a city, like just getting hit by a car. So I don't have the numbers whether their lifespan is longer or shorter. I would say a city raccoon could probably live if it's in the right spot. It can live a nice cushy life. Um,
Okay, so then where do raccoons live during the day when they're not foraging at night?
yeah, you can if you want to look for them. They might be in trees, um, nesting. Um, yeah, I mean, Sarah, you've probably seen a few like in the tree holes in Greenwood, just
Yeah,
hiding in the
my
daytime.
no, for sure. I mean, they can be in like the little crooks up in large trees. They can be in shrubs, in like underground dens. Um, there's another researcher looking at just sort of like the range of raccoons, and it seems like they have multiple dens. Like they just kind of like, stick it out during the day wherever they find themselves at the end of the night, which is kind of funny and cute.
Yeah, it's
Um,
very possible.
yeah. Um, let's see. Yeah, the rabies mitigation efforts are fantastic. Um, there has been rabies there's been positive cases up in the Bronx and like Queens, and I think two cases in northern Brooklyn at the end. Um, Lisa would like to know if we have any sense of raccoon populations at Greenwood in the past decades or past centuries.
I mean, I personally don't know the numbers. When people ask me, I always say there's a bunch.
There's a,
Like I just say over 200 maybe, but I don't know. So my cameras aren't really suited for telling population number. I can tell you that there's more raccoons in this area than another area where I have cameras, so I can compare. I can't really give you an exact number. I don't know. Maybe
yeah, and I will say I think raccoons move around a lot and they're really tricky to track. They're like they're very sneaky. Um, Hold on.
I will say though that Greenwood has, I think, the most skunks in New York City. I've yeah, I don't get a lot of skunks around Brooklyn aside from Greenwood, so I thought that was pretty cool.
Okay. Yeah, no, for sure. Debbie has a really good thought and question. She's a nurse and she's wondering if public education has been or could be developed and posted in public spaces in multiple languages about just like the animals that are rabies vectors and like the education around that. Um, I mean, first and foremost, even if the animal doesn't look sick, I always say give it, social distance is like what you should be doing. Um, but I'm not sure about the public education efforts from the city. Are you?
Yeah, I can say now that, you know, because this is such a recent development where like people are like, wow, there's wildlife here. It's a very recent thing. And Parks Department recently established a wildlife unit within the Parks Department, specifically because of this. And they do outreach education, but they're still a very new part of the Parks Department. And they're working hard to develop that material. You can go on their website, uh, and they have education campaigns. I'm sure they're planning on making their way out to schools as well. But you know, Parks Department is like aware of this increasing issue, and they've created a branch within their department to kind of gather information on this.
If inner city raccoons are bigger because they have, more food sources here, do you think that through your images you can sort of show that difference in size through the urban rural transect gradient?
Oh wow. You might have just given me another thesis question there. Yes, I could actually. I didn't really think to do that, but something you can do is you can put like a stick a certain distance away from the camera, and this can give you an idea of the size of something that turns up in an image because you can compare it to that stick that's in the image of your camera. And so if I do that with cameras along the transect, I can kind of look at how size is changing. You know, you need a lot of data to make that assumption, but that's a really good question. Wow.
Do
That's a
it
great
so you
question.
can answer the people. Um,
Yeah.
Magnus would like to know the similarities and the differences between a possum and a raccoon.
Oh, wow. So let's see. Do I have a okay? So if you look, the top is a possum, bottom is a raccoon, and one of the first things I notice when I see a raccoon is that the highest part of its body is the midsection, so it's kind of got an arched back. It's always looked like it's hunched over. The biggest differences are, I mean, they do look very different. A possum's face is long. It's long and it's got a white face. Um, and some people will think they're just huge rats because just from the backside, their tail is long and ratty. Um, Biggest difference I would say is, I mean, there's a lot that's different about them. Just the way they look. Um,
How about the way
Maybe
they reproduce? Like, why do we see more raccoons than we see, like, why did more raccoons come up than opossums?
yeah, that's a good question. I think possums actually are they're really okay with just being anywhere, and they like abandoned buildings. Um, and so you might see them, they're not really as focused in green spaces as raccoons might might be, because raccoons do like trees. They can go anywhere, you know, they can go in backyards and everything, but they tend to kind of, you know, need some trees at some point. But possums, you know, I see them like on the streets of Brooklyn. Like if you're out late enough, you might see them just walk by in the middle of the street. Um, and so I think they're just more tolerant and more spread out. So you might not get them as much on a camera trap in a green space because they're spread out. I can't say it's considered that possums and raccoons are both the most abundant. Maybe raccoons might be slightly higher, but it's probably just because people detect them more. I can't say for sure.
Um, yeah. Ooh, everything is coming in so fast. Yeah, Olivia says that possums are North America's only marsupial, I believe. Um,
Yes.
uh, hold on. Yeah, how many young can raccoons what's the offspring number? And then we might need to wrap this up to get this to Theodore? So what's like the reproduction rate and number of raccoons?
Uh, Let me think. So they usually give birth around springtime. Their litter size, I want to say, I'm gonna have to Google. I'd say 2 to 5, maybe 5 on the higher end. But I'm just gonna double check. Yeah, 2 to 5. I don't want to give you wrong information. Um, yeah, but they can reproduce each year. So
Oh, Mary Ting, you're breaking my heart. Mary says that opossums have short lifespans and can't survive well in our winters, except for now, because
it's a
we're not
wonder.
really seeing a winter. JC wants to know, and I feel like this is also a question of mine. Do you think there's a lot of migration of raccoons between Greenwood and Prospect Park?
Ah,
And
I
I think like the sort of like meat of this question is that Prospect Park and the cemetery have very different things going on. Like Prospect Park has a lot more foot traffic. The cemetery is closed at night, right?
mm,
And these are like nocturnal animals. So do you think, yeah,
it's a great question. Cameras can't really answer that, but fortunately, we have had a student that did put radio collars on some raccoons, and you do get the sense that they can kind of reach out there. I don't think it's extremely like every night they're just hopping between each space, but they can end up, they can move out to there. There can be some connectivity. I don't think it's like, you know, they're hopping over there all the time, but they can move uh to Prospect Park as well. Just not as frequently as you might think. There's not really a reason for them to. There's raccoons, there's enough trash in between Prospect and Greenwood for them to like not really want to go all the way over there. But it can
I
happen.
mean, there are a ton of restaurants that are all around these green spaces. Mary Ting just keeps breaking my heart. Opossums lose, they're often found with missing toes. Oh my goodness. Um, all right. And do you think that the camera trap methodology can be used for smaller wildlife like bats, nocturnal pollinators? Do you think the camera is sensitive enough to capture that?
Yes, yeah, it can do this. Depending on which your target group is, you might want to set up the camera differently. I have these knee height. If you're looking at bats, you want to set them up near maybe some kind of bat roost or some attractant source higher up. Um, they're not really good for things like mice and chipmunks. You literally have to turn the camera towards the ground to catch them reliably. So, it's you got to change positioning for different groups of species. And yeah, but you can use them for birds, bats. People do use them. I'd say for these mid-sized mammals, camera traps are ideal, but they can and have been used for other species as well.
All right. Last question is mine. I'm not sure if you've assessed this, but what are some of the patterns of distribution that you have found?
Yes. So it's hard to say right now because we have a lot of data to go through, but I have generally found, it's exactly what you'd expect with coyotes and foxes is that, we're seeing more of them out Nassau, maybe beginning of Queens, and not really any in Brooklyn yet. Uh, the skunks is pretty interesting because, like I said, you have complete dead zones, and like I might only see skunks in Greenwood, uh, or one other site. So their distribution is very like irregular. Um, for the raccoons, I need to actually collect more data to find a pattern because right now it looks like they're just kind of everywhere.
Interesting.
um, yeah, so it's hard to tease out which variable might be influencing their distributions the most. What else is there? Yeah.
Everyone, okay, this is last two-part question, and then we should move on to Theodore. How much are the camera traps and where can they get them?
Yeah. Well, if you want to get them in person, B&H, there's a few B&H's in the city. It's like a video equipment store. You can get them there, discounted actually. You can get them on Amazon. Uh, a small one like this, I think this is like a mini micro one. I think it's $60 to $70. And they could be anywhere from the ones I use are $110. You can get them cheaper. So yeah, they're affordable. And it's really great to, if you want to set one up in your backyard, they're really easy to operate. You just use an SD card, and yeah, it's a fun thing to play around with.
Well, I request that you drop a link in the chat box for people to shop around. And
Yes.
then if you can stop sharing your screen, let's pass it on over to Theodore.
All right, good. Can everyone see my screen there?
Yep,
Everyone can hear me all right?
yep, perfectly.
All right, great. So thank you all for tuning in this evening. And thank you to Greenwood for giving me an opportunity to talk about the research that we've been carrying out over the last 8 months or so. I live just a couple of blocks away from the cemetery, and spent a lot of time walking through the cemetery and bird watching in the cemetery. So I was really super excited to get this award, to be able to actually carry out some research in the cemetery as well. My background, I'm a microbiologist at Brooklyn College, and one of the main interests of my group is to study our urban microbial communities. These urban microbiomes, we're quite interested in how human activity influences the composition of urban microbiomes. And we're interested in how we might be able to tweak the microbiomes or alter them so that they are more efficient at providing some important services like decomposition, bioremediation, carbon capture, and some other ecosystem services. So that's kind of encompasses a number of projects that are ongoing in the lab. And today I'm just going to talk about the work that we've been doing in Greenwood. And here the primary question is that we wanted to look at how management intensity within the cemetery might impact bacterial communities. Whoops. Let me see if I can get this to advance. Oops. There we go. So I'll just introduce the group that's been working with me at Brooklyn College. A number of students have worked. We did 2 collections this past year, one in July and one in October. And I just wanted to draw a particular note to Shelby Luster over here, who led most of the efforts in collecting samples in July, and Irene Lesyuk, who led most of the collections in October, and Alonso Cordoba, who's been working with me for several years on a number of soil projects across the city, and he also was a big part of the collection at Greenwood Cemetery. I wanted to also just again thank Greenwood Cemetery for the support to carry out this work. And I also wanted to thank the NSF. There's a summer research program. A colleague of mine in the biology department, Tony Wilson, runs the Brooklyn Undergraduate Environmental Ecology Program. And this is a program that college students from across the country can apply for. And both Shelby and Anna here were supported by this BUI program. And so Shelby is based out of Southern California at the Scripps College. Anna's from Manhattan, and she's attending college at Wellesley. So these are the people that did the real work. So let me just give a little background. As probably everyone is aware, soils contain billions of microorganisms. So just loaded with all sorts of good microorganisms. And we consider all of these together, any of the microorganisms in a given sample would be the microbiome. And so this will be the bacteria, fungi, viruses, protozoa. All of that is what we're going to refer to as the microbiome of a given sample. And again, as you can imagine, soils are not uniform. They can vary greatly in their physical, chemical composition, and even on small spatial scales, there can be significant differences. And these differences create many different micro niches that can support a wide array of different microorganisms. And I love to show this picture here in the upper right-hand corner. This is a grain of sand that's been examined under the microscope, and all of the bacteria growing on this grain of sand are stained in green. So all this green is representing bacteria that are growing on that grain of sand. And as you can see, they're not uniformly distributed. They're in these little pockets and crevices. And there are other images from this same paper where they've actually colored the bacteria based on their phylogenetic background. And so if you look at those images, you can see that it's not even the same types of bacteria colonizing this grain of sand. So if you can have all that diversity just on a grain of sand, you can imagine extrapolating to the types of diversity that you would have in a complex soil, such as those in Greenwood Cemetery. And of course, there are other things that influence the number and types of bacteria in the soil. Plant roots, that's the rhizosphere. Plants and trees turn out lots of chemicals and carbon sources that bacteria and other microorganisms like to utilize. Worms and other insects can modify the environment and also create specific niches for microorganisms. If you look at this central figure, if you look at a kind of a gram per gram basis in the soil, the majority of microorganisms there are going to be bacteria and fungi. Archaea, which are quite similar to bacteria, also are prevalent in the soil, and then the protists and the viruses make up, again, gram for gram, a smaller number in the soil. And today I'm going to focus mostly on the bacteria. So as I might have alluded to, these soil microorganisms contribute to and drive a lot of the important ecosystem services that are provided by soils. And they contribute, as I said, to decomposition, nutrient cycling, mineral availability. They can suppress pathogens. They can help with water retention and other processes. And I'm not going to go through all the kind of chemical cycles here, but this is just one figure from a review that kind of illustrates the number and variety of different chemical processes that are going on in the soil and that are driven by the microorganisms in the soil. I also, again, just talked about all the different niches, and of course the physical factors and environmental elements within the soil can vary quite a bit. And here on the lower left is a list of some of the most, the major features that can dictate the composition of the microbial community within soils. Okay, and it goes from the left-hand side being those factors that have less influence, like predation and plant species and temperature, over to the right to the factors that are more strongly associated with driving the composition of these soil microbial communities, and that's pH, carbon availability, redox state, and moisture. And not shown here, but human activity also can alter these factors and can therefore have a major impact on the soil microbial communities. And in a number of the studies that we work on in the city, we oftentimes categorize soil samples into two different types. Those that are naturally deposited. So these are going to be soils formed in place by processes like glacial activity, sediment deposition, wind, erosion, and so forth. So those are the natural soils. And then the other category we call human altered and human transported materials. And these are going to be soils that are strongly influenced by human activity. They can be fill, they can contain construction debris and other waste. They oftentimes are extensively managed. They can be transplanted soils in the cases of green infrastructure like tree pits and green roofs and bioswales and so forth. And as Myles mentioned in his talk, there's lots of buildings and lots of streets and lots of sidewalks in the cities. So the estimate is that in New York City, about 63% of the surface is impermeable. 28% of the soils that you can see fit into this human altered and human transported category. And just a little under 10% are actually from this naturally deposited material. And when we look at Greenwood Cemetery, most of the soils that are in the cemetery are going to be originally from this naturally deposited category, but because it's an active cemetery, there's going to be a large degree of human influence on those soils. So I'm just going to kind of give a little overview of the experimental design for this project. We categorized 3 levels of management intensity for this project. And so if you look down here at the bottom, we have the managed areas. We have then an intermediate level of management, which are referred to as these perpetual meadows. And you can see they're kind of have a meadowy characteristic. And then the third category are minimally managed woodlots that you can see here on the right. And we identified 3 sites, 3 from each level of management. So for a total of 9 sites. And then in each of those 9 sites, we identified 2 different positions. And from each of those 2 positions, we collected soil samples at a shallow depth of 5 centimeters and a deeper depth of about 20 centimeters. And so for those 9 sites at 2 different locations and 2 depths, where 36 samples were collected, and we did this once in July and once in October. So we have a total of 72 samples that we were dealing with in most of these analyses. And I've kind of mapped them out on this map here. This is a map from Greenwood, again, showing different areas of different management, and was this map that we worked with in terms of trying to select those sites that we collected soil from. So I'll take a minute. I'm not going to go into a lot of the details here, but this is the basic metagenomic strategy that we use to analyze soil microbial communities. So here is my not particularly inspiring diagram of a complex microbial community, which could be the soil, it could be the gut, could be the skin, could be a water body, any place where you're going to have a complex mix of microorganisms. And each of these species has their own genome, and that genome has distinct sequences of bases, these A, Cs, Gs, and Ts that make up the DNA. And so each species can be specifically identified by unique stretches of DNA. And in the case of bacteria, which is what we're going to focus on in this study, we target a particular gene called the 16S ribosomal RNA gene. This is present in all bacteria, so we know all bacteria have this gene, and we use this targeted amplicon sequencing approach to sequence those 16S genes to identify the different types of bacteria in our sample. There are other methods like shotgun metagenomic sequencing, but we didn't use that in this project. And so if you look at the flow for this project, or the kind of the analysis pipeline, we start with our complex soil community, which we're going out into Greenwood and collecting. In the lab, we extract the DNA from those soil samples, and we don't need a whole lot, just a couple of grams. We take that purified DNA and we send it to a sequencing company. The one that we're using is called Wright Labs in Huntington, Pennsylvania. And so they'll take our DNA samples and sequence them on the Illumina platform, and they give us millions of sequence reads of that specific 16S ribosomal RNA gene back. We then take those raw sequence data that are delivered to us by the sequencing company, and we run them through an analysis pipeline called CHIME, which is quantitative insights into microbial ecology. And what CHIME does is it looks at each one of those sequences, and each of these sequences is about 300 base pairs long, and it compares them to the database, and it says, well, first it categorizes it, so it bins them. So everything that's within, 97% identical, you can set the threshold, but 97% is pretty typical. Everything that's 97% similar gets binned into one group. And so it goes through all the millions of sequences and creates hundreds or thousands of bins with very, very unique sequences. And then it compares those sequences to the database. And based on the sequence, you can get a determination of what bacterial species is likely represented by that sequence. And so you get specific information on the composition, as well as by counting the number of sequences that go into the bin, you know that, okay, in this case for CGGTAA, we got 28 counts. And for this other one, TTAAAC, we only got 5. So we get both information on the type of bacteria and also the relative abundance. And so again, just in a nutshell, we're using the DNA as a proxy for the microorganisms that are present in the soil. And so if we look at the data from Greenwood Cemetery, if this is a figure that shows all of the data from all of the sites that we collected from, put together. And what we did was we made a rank abundance curve. And so on the x-axis down here, you can see the different bacterial taxa that were present in our sample. And on the y-axis is the number of times we saw that sequence in the sample. So if you're way over here on the left, in this case, the most abundant one, we counted somewhere between 25 and 30,000 times, okay? And then it starts to drop off, and you can see that there's this extremely long tail, which actually the figure would kind of have to go off several more screens to cover the entire slightly over 1,000 taxa that we were able to identify. But what I want to draw your attention to is that there are a few highly abundant taxa, okay, and then hundreds of bacterial types that we detect very infrequently. And this is typical. If you were to look at the microbiome from your gut or your skin or from most other samples, and you made a rank abundance curve, you would see something very similar to this. There are tons of diversity, lots of different bacteria, but a few of them tend to be very abundant. Most of them are very infrequent. And so in this case, I took the top 10 that we encountered, and I'm listing them here. I won't go into this, but I'll just say that, I'm a microbiologist and I don't recognize most of these. So there's a lot that we don't know. We're not coming up with, all the common bacteria that you see. These are ones that are, I looked them up. There's maybe 1 or 2 publications on them, very, very little. And so it's really kind of an exciting world because there's so little known about these bacteria. If we look at these data in a slightly different view, here, these are 100% abundance bar charts. And here along the x-axis in this first graph on the left, these are the 9 different sites. And on the y-axis is the relative abundance. So it's like what percentage of the total did a specific phylum contribute to, okay? And so the phyla here are color-coded. Now, with our sequence data, we can oftentimes get down to the taxonomic level of genus. But then you get this, crazy rainbow of colors that's just too busy to really look at. So typically we stick at these higher taxonomic orders like phyla, and we kind of compare our samples at those levels. And what you can see is that in these soils for all of the 9 sites, there are 3 very abundant phyla. There are proteobacteria, make up about 1/3. Then there are acetobacteria and actinobacteria. And together, those 3 phyla, alone, make up about 70%, 65 to 70% of all of the sequences that we saw. And then there's another dozen or so phyla that make up the remaining 35 to 30%. And again, this is typical of soils. These 3 phyla are the most abundant phyla in pretty much all soils that have been studied to date. So that's typical. If you look to the lower right-hand side, now I've grouped all the samples by management. So on the left is the managed, in the middle is the perpetual meadow, and on the right is the woodlots. And again, when you look at the 3 at this level, at phyla, they look very similar. There aren't any significant differences. However, if you look up in the upper right-hand corner, here I've grouped on the left all the samples that were collected from 5 centimeters compared to all the samples that were collected at 20 centimeters. And here you can see that there is an increase in the acetobacteria at the deeper depths and a decrease in the actinobacteria. And so that you can have the differences. Again, it depends on how you look at your samples, but depth, does have an influence on the community composition, whereas at the individual sites and the management, using this method doesn't show a significant difference in the community composition. I'm not going to spend a lot of time here, but these are the 3 major phyla that show up. The proteobacteria include lots of things that you might have heard of, like E. coli. A lot of human pathogens fall into the proteobacteria group. Actinobacteria include streptomyces, which the group of streptomyces make a lot of the antibiotics that we use. And then the acetobacteria, they kind of have diverse metabolism. They're comfortable in a range of environments. And again, they make up a major portion of the soil bacterial community.
Hey, Theodore, I have a quick question.
Yeah.
What is the difference? What's gram-positive and gram-negative?
Oh, yeah. Okay. So for the most part, there'll be some exceptions to this, but the proteobacteria are going to be gram-negative. The acetobacteria are going to be gram-negative, and the actinobacteria are going to be gram-positive. But from a functional standpoint, gram-positive and gram-negative bacteria can do a lot of the same things within the soil.
What does it mean though?
Oh, what does it mean?
Yeah.
Okay, so it's based on a microscopic observation that the gram-negative bacteria have a relatively thin cell wall, and the gram-positive bacteria have a thick cell wall. And so when the staining technique, the gram stain, was developed, it utilizes a dye called crystal violet. And when you stain, the crystal violet stays trapped in the gram-positive bacteria because that cell wall is so thick that the crystal violet dye can't be washed out easily. Whereas in gram-negative cells, that crystal violet dye gets washed out quite easily. And actually in the original stain, those gram-negatives are colorless, but then a modification to the stain adds a second dye afterwards that turns those gram-negatives pink. So when you look under the microscope, if you've done a typical gram stain, the gram-positive cells are purple, the gram-negatives are pink. And the important difference there is the structural differences. The gram-positive with this thick cell wall, the gram-negatives with a relatively thin cell wall.
Gotcha.
But again, functionally, they can do a lot of the same things.
Gotcha. Thank you.
I can come back to this later if people are interested. This is a look at beta diversity, but for time, I'll skip this one for a minute. So what I showed you in the last couple of slides was based on this sequencing approach, using the DNA sequences as a proxy for the microorganisms that are present there. We also used what are called eco plates to look at the carbon utilization profile of these soil samples. And so in this approach, we use these 96 well plates, and these wells are pre-loaded with 31 different carbon sources, and it's in triplicate with a water control, so it adds up to 96. And so what we do is we take our little soil sample, we mix it up in water, and then we add, here you can see the samples being pipetted into the 96 well plate. And then we incubate them for 2 days in the dark at room temperature. And if there are bacteria in that sample that are able to utilize the carbon source, then you get this color transformation. So you get this blue to purple color transition. If the bacteria in the community are not able to utilize that carbon source, then it remains mostly clear. And then we can put this into a machine called a plate reader that gives us a quantitative measurement of how much of a color change was there in each sample. I've made a heat map of all the data that we collected, and you can see that, actually I'm blocking part of my own screen, but the compound tween 80 had the highest utilization from all the soil samples. L-serine used as a carbon source had the least usage. But what's more interesting is that when we look at the metabolic activity of these different samples, and again, here on the x-axis, we have the different sample types, and on the y-axis is a measurement of the overall metabolic activity, or the overall ability to utilize those 31 carbon sources. So the higher you are up on the y-axis here, the more utilization of those carbon sources that sample was able to do. And we can see some seasonal differences, and we can see some depth differences. So if you look here on the far left is July at 5 centimeters, and in the kind of greenish yellow is July at 20 centimeters. And these little bars represent the average. And so you can see that at 5 centimeters, we get quite a bit more carbon utilization than we do at 20 centimeters. And if you look at, here's July, and October is in red, and I guess this purpley color. And again, you see the same difference in, more activity at 5 centimeters, less at 20 centimeters, but you also see, compared to the summer samples, there's less activity in the samples collected in October. And then if we go down to the lower right-hand side, what I think is really cool is that when we look at the management, now remember, for management, we didn't really see a whole big difference when we looked at the composition of the microbial communities or the bacterial communities using the 16S approach. Here, I've grouped, this is all at the 5 centimeter depth, because I only wanted to compare one variable. I didn't want 20 centimeters to kind of complicate things. But if you look at managed and perpetual meadow and the woodlots, this bar represents the average. And the average metabolic use or activity is about the same in all three management regimes. But what you can see is that with management, they're all kind of tightly clustered around the mean. At perpetual meadow, that cluster kind of spreads out a bit, even though the mean is the same. And in the woodlots, you really get a lot of spread, even though, again, the mean is about the same for all three. So what that suggests is that the variance is inversely related to the intensity of management. So the more managed the sample is, the less variance, and the less managed, you have much greater variance. So that, you might predict that, but it's really nice to see that show up in the data that we have here using these eco plates. And so that's, we've got tons and tons of data. We're still really actively working on and analyzing more of these data. But that's where things stand at the moment. And I can just, summarize to say that the soils are dominated by the proteobacteria, acidobacteria, actinobacteria, and that's typical. There is some a difference in composition by depth, and that's also expected because we and others have seen that in other soil samples. The management practice doesn't seem to change the soil composition, the bacterial composition significantly, but we do see this effect on variance that with more management, we see less variation. And I will again thank you for your attention, and thanks to Greenwood for giving me the chance to talk about this work.
Great. So Logan is asking, was there anything that surprised you in your data that you hadn't seen in any of your prior study of soil samples?
Well, like I said, we're still looking. This was the first project that we used the eco plates with, and so we were kind of excited to see these data. The thing to keep in mind about using the DNA as a proxy for the community is that DNA can stick around a while after the bacteria die, and it's also going to be there in bacteria that might be dormant or spores. And so, it's you have to kind of take that into consideration. Whereas the eco plates are dependent on, those bacteria have to be growing and metabolizing that carbon source in order to see that color change. And so that's really going to be a reflection of the bacteria that are in the sample that are really alive and able to grow. And so we were glad that we could add that as an additional factor. But one of the things that we do typically see in city soils, and this was a little counterintuitive at first, but we've seen it multiple times and colleagues have seen it in other samples, is that roughed up, kind of nasty looking soils in the city actually have super high diversity. And we think that might be because, all the human influences actually create more micro niches and give space for more different types of microorganisms to grow. That's not necessarily a good thing. We don't know that those bacteria are all doing positive things. Many of them are probably not. But I think generally people think of like increased human activity and lower biodiversity. And in this case, it's increased human influence and possibly increased biodiversity, at least at the microbial level.
We have another question, but I have a question for you to sort of further explain what is the conversion to biomass carbon, and if it happens more higher or in the shallow profile of the soil, what does that mean?
So if I understand the question, so we didn't measure anything where we would be able to say this amount of carbon was utilized. What the eco plate tells us is it gives the bacterial community a choice of these 31 carbon sources, and it basically gives you a sense of how much range do they have in using a whole lot of different possible sources, but it's not giving us values on how much total carbon was utilized. So we didn't do any studies that would kind of give you those numbers, but other people have done studies. And if you just say that our soil samples are similar to other soil samples, then you could use that carbon data from others. But we didn't specifically collect that.
For sure. Adrian is curious about the relationship between landscape management and carbon sequestration in soils. So what would an additional study of the microbial communities tell us about a soil's potential for holding more carbon?
Right. Well, it's an excellent question, and those are the things that we're kind of looking at or looking forward to doing in the future. But again, the work that we did here doesn't allow us to address that question. But what I can say is that, from these three different management profiles, the microbiomes are diverse. They're similar to healthy soils that we've seen elsewhere. And so I think that in terms of carbon utilization, it's, there are plants and trees that you can plant that are going to sequester more carbon, say, than a grass sod. And the microbial communities, they look great. They look like whatever you put there is probably going to do fine. There's not going to be some inability of some types of plants or trees not to grow because the microbial community is not going to support that. But I think to a certain extent, yes, there could be differences in how much carbon is sequestered or used by the microbial community, but I think it's a lot of that could be, if you really wanted to have an impact, it's going to depend on what you grow on that soil in terms of cover.
Yeah. Are there differences from greenwoods, wooded lots, their soils, and wild forest wild forest soils? There we go.
We didn't specifically compare the greenwood wooded lots to other wooded lots, but we, in a similar study, we used as a undisturbed control soil from a forest in a state park in New Jersey. And again, here was kind of the counterintuitive thing, is that our urban soils had a greater biodiversity than this kind of pristine wooded forest area in New Jersey. And so if I were to guess, I would say they'd probably be pretty similar, but there might be slightly greater diversity in the greenwood woodlots. But again, we don't know if that's good or bad. So you kind of have to, just keep that in mind.
The variation in diversity, would it be more different in someone's backyard than what we were seeing at Greenwood? Or at the parks?
So more different than, so I'm not sure I fully understand, but one thing to keep in mind is that these measurements of diversity are based on like how many different taxa do we count, and there's different ways to do that. So just because we see 100 taxa in one sample and 100 taxa in another sample, you could say that they are equally diverse, but it could be a completely different mix of bacteria. It wouldn't have to be the same. Yeah. The other thing that we often look at is evenness. So you could have a soil that has 100 bacteria, but like we see here, 90% of those bacteria come from just a handful of those 100 species. Whereas in another more even sample, you could have 100 species, and each species makes up 1% of the total abundance. That's like an evenness of 1. It's perfectly even. Each different species is abundant at the same level. So these soils tend not to be, well, soils in general tend not to be that even. You have a few groups that make up the majority, and then hundreds that make up, have relatively small contributions. And again, the question for soil and question for the gut microbiome and any other microbiome that's been studied is how important is that long tail of diversity? Does it really matter that there's, hundreds of species there at very low levels? Are they important for the health of the soil, for the health of your gut? Those are questions that are actively being addressed.
Yeah. What is the significance of more bacterial variation in the soil? So I guess like in terms of the significance of urban soils.
So in the case like where we were, I don't know if I can flip back here, but in this case where if you're talking about the variation, what's the importance of that? It just means that, in this case, that if you go to a wooded lot and you go to say plant something in a specific spot, or you have some expectations about what the quality of the soil and what the community is going to be in a particular spot, you might be very wrong, because you might be looking at one of these points high up on the y-axis or one of these points low down on the y-axis. So there's a lot more unknown from one spot to another, even though the average comes out to be about the same. Whereas in those sodded, kind of grass areas, the managed areas, pretty much any place you go, you're going to get a pretty consistent finding on the bacterial composition there, and probably other soil properties. So I'm not sure if that answers the question, but that's kind of like the practical importance of the variance, is that when it's very small, then any sample you look at is probably going to be pretty close to the mean. Whereas if the variance is large, then you could be anywhere on this range when you look at a specific point.
Gotcha. And then I think I can answer Catherine's question, because you weren't looking for human DNA, but if you found traces of human DNA in the soil. And then she apologizes that she's asking.
Well, I'm sure we would, because we use the 16S amplicon sequencing, humans don't have, in your own cells, you don't have the 16S gene, so we wouldn't detect it. Your bacteria do, so we could detect human bacteria. But again, there's overlap between soil bacteria and human bacteria anyways. But you certainly, if you use the other technique, if you remember back to one of the earlier slides, the whole shotgun metagenomic approach, that is not focused on a specific gene. And in those cases, you very well could detect human DNA, but it would probably be buried in there with lots of plant DNA and worm DNA and bacterial DNA and fungal DNA and viral DNA and whatever. So my expectation is that it would not make up a significant fraction.
Yeah, I think just the direction of this question, I do want to remind that like our DNA is kind of shedding from us all the time, like with our skin cells and our hair and, you know, just, yeah. All right. Any plans for Greenwood to employ green burial? Yeah, green burial is available right now. That was a question to Aaron. All right, so we are down to the wire. Are there any last questions or comments? All right, Theodore and Myles, thank you so much. It has been such a pleasure to work with you, and we have a little bit more work to do, but thank you so much for being with us tonight. Thank you everyone for joining in. And we will see you later. This will be recorded, by the way, and we will post it likely on YouTube or somewhere on our website. All right, have a good night.